An allele, or allele, is one of a number of alternative forms of the same gene or same genetic locus. It is the alternative form of a gene for a character producing different effects. Sometimes, different alleles can result in different observable phenotypic traits, such as different pigmentation. However, many genetic variations result in little or no observable variation. Most multicellular organisms have two sets of chromosomes. That is, they are diploid. These chromosomes are referred to as homologous chromosomes. Diploid organisms have one copy of each gene on each chromosome. If both alleles are the same, they and the organism are homozygous and the organisms are homozygotes. If the alleles are different, they and the organism are heterozygous and the organisms are heterozygotes. The word allele is a short form of lemorph, which was used in the early days of genetics to describe variant forms of a gene detected as different phenotypes. It derives from the Greek prefix =iii registered trademark I, allele, meaning reciprocal, or each other, which itself is related to the Greek adjective =iii, meaning other. Dominant and recessive alleles, in many cases, genotypic interactions between the two alleles at a locus can be described as dominant or recessive, according to which of the two homozygous genotypes the phenotype of the heterozygote most resembles. Where the heterozygote is indistinguishable from one of the homozygotes, the allele involved is said to be dominant to the other, which is said to be recessive to the former. The degree and pattern of dominance varies among loci. For a further discussion, see dominance. This type of interaction was first formally described by Gregor Mendel. However, many traits defy this simple categorization and the phenotypes are modeled by polygenic inheritance. The term wild-type allele is sometimes used to describe an allele that is thought to contribute to the typical phenotypic character as seen in wild populations of organisms, such as fruit flies. Such a wild-type allele was historically regarded as dominant, common, and normal, in contrast to mutant alleles regarded as recessive, rare, and frequently deleterious. It was commonly thought that most individuals were homozygous for the wild-type allele at most gene loci, and that any alternative mutant allele was found in homozygous form in a small minority of affected individuals, often as genetic diseases, and more frequently in heterozygous form in carriers for the mutant allele. It is now appreciated that most or all gene loci are highly polymorphic, with multiple alleles, whose frequencies vary from population to population, and that a great deal of genetic variation is hidden in the form of alleles that do not produce obvious phenotypic differences. Multiple alleles. A population or species of organisms typically includes multiple alleles at each locus among various individuals. Allelic variation at a locus is measurable as the number of alleles present, or the proportion of heterozygotes in the population. For example, at the gene locus for the ABO blood-type carbohydrate antigens in humans, classical genetics recognizes three alleles, IA, IB, and E, that determine compatibility of blood transfusions. Any individual has one of 6 possible genotypes that produce one of 4 possible phenotypes, type A, type B, type AB produced by IAIB heterozygous genotype, and type O produced by E homozygous genotype. It is now known that each of the A, B, and O alleles is actually a class of multiple alleles with different DNA sequences that produce proteins with identical properties. More than 70 alleles are known at the ABO locus. An individual with type A blood may be an AO heterozygote, an AA homozygote, or an AA heterozygote with 2 different A alleles. Allele and genotype frequencies. The frequency of alleles in a diploid population can be used to predict the frequencies of the corresponding genotypes. For a simple model, with 2 alleles, where P is the frequency of one allele and Q is the frequency of the alternative allele, which necessarily sum to unity. Then, P2 is the fraction of the population homozygous for the first allele, 2pq is the fraction of heterozygotes, and Q2 is the fraction homozygous for the alternative allele. If the first allele is dominant to the second, then the fraction of the population that will show the dominant phenotype is P2 plus 2pq. And the fraction with the recessive phenotype is Q2. With 3 alleles, and in the case of multiple alleles at a diploid locus, The number of possible genotypes with a number of alleles is given by the expression. Allelic variation in genetic disorders. A number of genetic disorders are caused when an individual inherits two recessive alleles for a single gene trait. Recessive genetic disorders include albinism, cystic fibrosis, galactosemia, phenylketonuria, and Tay-Sachs disease. Other disorders are also due to recessive alleles, but because the gene locus is located on the X chromosome, so that males have only one copy, they are more frequent in males than in females. Examples include red-green color blindness and Fragile X syndrome. Other disorders, such as Huntington disease, occur when an individual inherits only one dominant allele. See also: allozyme, evolution, genealogical DNA test, haplosufficiency, meiosis, Mendelian error, Mendelian inheritance, mitosis, polymorphism, Punnett square. References and notes. National Geographic Society, Alton Biggs, Lucy Daniel, Edward Ortalb, Peter Ilero, Dinah Zeke. Life Science. New York, Ohio, California, Illinois, Glencoe McGraw-Hill 2002. External links: Alfred, The Allele Frequency Database, straft.org. Earth Human Short Tandem Repeat Allele Frequencies Database.